My name's Tom Titus, and I'm a research associate in John Postlewaite's lab in the Institute of Neuroscience. One of the big projects that we do in the lab is a mutation screen, and we target specific genes, target a particular amplicon from a specific gene that's likely to give us a mutation that will give a phenotype in the end. And we've developed a method where we can mutagenize a line of fish and screen the heterozygous outcross, approximately 8,000 fish, and screen those by PCR and actually identify heterozygous carriers for knockout mutations. We initially quantitated 2,000 of our DNA samples using some standard absorbance technology, and that ended up costing us about 6 months actually of time on our mutation screen, simply because the DNAs weren't accurately quantitated, and when we normalized them, it was they were unusable. What ended up happening was we had fish that were the genomic DNA that was over diluted compared to other samples in the screen, and so essentially we lost all those fish to our screen. We weren't able to identify any mutations that any of those fish would have been carrying, and it took us quite some time to figure out that that was the fundamental problem in our screen during that time. So we changed to the Qubit so that we could get our DNA concentrations really nailed down. What we ended up doing was going back and using the Qubit to measure all of those samples that we thought we had normalized in the beginning. And of course what we discovered was there was at least a 10-fold difference across samples in terms of DNA concentration. And that's partly because I think we were pushing the limits of the previous technology. Some of our DNA concentrations were low, probably too low to be accurately measured, and whereas the Qubit, we have lots of flexibility in terms of being able to use either a high sensitivity kit or a broad range kit to measure a diversity of concentrations accurately. Oh yeah, we do recommend it. And we recommend that people use our machine, and we do it for 2 reasons. One, it's well, 3 reasons really. It's accurate. That's the main thing. We can believe the results. We recommend it because it's easy to use, and we recommend it because the upstream cost of using the machine in terms of reagents is really inexpensive compared to the downstream cost of not having DNA accurately quantitated. It takes to run 1 lane of paired end sequencing on our next-gen sequencer. Our cost here is $2,000. So that's a lot of money.
Right.
Compared to a few bucks on the upstream side to make sure the DNA is accurately quantitated.